Welcome for today's session. Today we will be starting the discussion with parasitology. So today we'll begin with introduction to parasitology. So at the end of the discussion, we will be able to understand the meaning of certain important terms in parasitology. Then we would be able to understand the dynamic relationship between host and parasite. We would understand the importance of studying the life cycle of parasites. We would be able to understand the nomenclature of parasites, the importance of studying the parasitic infections, the zoological classification of parasites, the methods employed for diagnosis and prevention of parasitic diseases. So let's begin with a brief introduction. So, man and other living things on earth live in an entangled relationship between each other. So, humans and the other living beings have some relationship with each other. They do not exist in an isolated fashion or in an isolated form. They are interdependent on each other. Medical parasitology is the stream which deals with organisms or living beings or the living creatures in or on the human body or the host and the medical significance of this host parasite relationship. This is a struggle for existence, or this is actually the struggle of existence which has led to some species of life forms to be dependent on the others, and in this the origin of intimate relationship between these species has evolved. Coming to what is predation. So predation is a relationship between two species where members of one species consumes the members of another species for its food. The predators are usually carnivorous animals like lion, tiger, and the preys are the herbivorous animals like the deers and the zebras. Coming to what is symbiosis. So symbiosis is a close living arrangement between two species. The word comes from Greek word which means state of living together. Symbiosis is classified into mutualism, commensalism, and parasitism, and these relationships range from beneficial to harmful relations between the two associations. Coming to mutualism, in this type of association, both the species are benefited. One of the best examples of mutualism is the relationship between the nitrogen-fixing bacteria called as rhizobium and the leguminous plants such as the beans. These rhizobium species, they fix the environmental nitrogen for the plants. The US biologist Lynn Margulis has postulated that eukaryotic cells evolved from the colonies of mutualistic bacteria. This theory is now widely accepted and is known as endosymbiosis. Coming to commensalism, this is a harmful relationship between two species where member of one species called as commensal gets the food, shelter from other called as host. In this relationship, one gets benefited, but the other neither is benefited nor is harmed. For example, the normal skin flora of humans. Coming to parasitism, this association is between two species where one is usually smaller, called as parasite, which derives food and shelter from the other, which is called as the host. In this relationship, the parasite causes injury, affecting the well-being of the host, and the host tries to curtail the damage and eliminate the parasite. Parasite in Greek means parasitos, which is eaters at the court, that is meat teasers. Three major groups of parasites infecting humans are protozoa, helminths, and arthropods. Coming to the systemic classification, the general scheme of classification includes the empire, kingdom, phylum, class, order, family, genus, and the species. The parasites have Latinized names and hence they are written in italics. These names consist of two scientific names. One part is the genus name and the other part is the species name. For example, Entamoeba histolytica, where Entamoeba is the genus and Histolytica is the species. The genus name always begins with a capital letter, while the species is in small letters. So here we can go through the taxonomic classification of protozoa. Then we have the taxonomic classification of helminths. Coming to types of parasites. So parasite is a living organism which lives in or on the body of other organisms, which is called as host, and derives the required life support in the form of food, nutrition, shelter directly from the host, and in return it does not give any benefit to the host. Protozoa and helminths are the studied parasites in medical parasitology. Parasites can be classified as ectoparasites. Ectoparasites inhabit the surface of host body without penetrating inside the host body or into the tissues. These are important vectors in transmitting the pathogenic microbes. Infection of these parasites is called as infestation. For example, the Sarcoptes scabies, which is an ectoparasite, causing scabies. Next, coming to endoparasites. These parasites live inside the body of the host. For example, the malarial parasite. Invasion by endoparasites is called as infection. The endoparasite infect the host while ectoparasites infest the host. Parasitic infections and infestations cause parasitic diseases. The endoparasites are of the following types. Obligate parasites. These parasites cannot exist without a parasitic life in the host. They cannot exist freely outside the host. For example, Plasmodium species. Then we have facultative parasites. These can live in a parasitic life as well as they can exist as free living forms. For example, the Acanthamoeba and Naegleria fowleri. Then the accidental or incidental parasites. These parasites infect an unusual host. For example, Echinococcus granulosus will accidentally infect humans. Then the erratic parasite. These are the one that wander into an organ in which they are not supposed to be found. For example, Entamoeba histolytica will be found in the liver or lung of humans. Aberrant parasite or wandering parasite. These parasites infect a host where they cannot live or develop further. For example, Toxocara in humans. Coming to host. Host is defined as an organism which harbors the parasite and it provides the parasite with shelter and nourishment. There are different types of hosts. For example, the definitive host. This is the host in which the parasite multiplies sexually or the adult parasite replicates sexually. For example, in malarial parasite, sexual multiplication takes place in female Anopheles mosquito. So female Anopheles mosquito is the definitive host of this parasite. Some protozoan parasites like Trypanosoma brucei, responsible for causing a disease which is called as sleeping sickness in humans, multiplies in a tsetse fly that transmits the disease to humans. In such situation, the mammalian host, that is humans, is the definitive host, and the vector, okay, the arthropod host, that is the tsetse fly, is an intermediate host. In helminths, a definitive host is the one which harbors the adult form of the parasite, while the intermediate host is the one that harbors the larval form. For example, humans are definitive host in tapeworm infections, for example, the Taenia species. Humans are again definitive host for many of the parasitic diseases. Coming to intermediate host, in helminths, a definitive host is the one which harbors the adult form of the parasite, while the intermediate host is the one which harbors the larval form of the parasite. The intermediate hosts are essential for completion of life cycle of some parasites. Some parasites require two intermediate hosts to complete their life cycle. These are known as the first and the second intermediate host respectively. For example, we have a parasite called as Fasciola hepatica, in which the first intermediate host is an amphibian snail and the second intermediate host is an aquatic plant. Hosts can be of different types, like the reservoir host. This host harbors the parasite and serves as an important source of infection to the other susceptible host. For example, dog is the reservoir of Echinococcus or Echinococcus granulosus. Then we have paratenic host. It is the host in which the parasite lives, but it cannot develop further and not essential for its life cycle. This is also known, this is known as the paratenic host. For example, we have a freshwater prawn for Angiostrongylus and a big fish for the pleurocercoid larva of Diphyllobothrium latum. Then we have something called as the amplifier host. It is the host in which the parasite lives and multiplies exponentially. Then we have the natural host. It is the host which is naturally infected with certain species of parasite. For example, pig is the natural host for Balantidium coli. Then we have accidental host. This is the host in which the parasite usually is not found or usually is not supposed to be seen. For example, man is an accidental host for Echinococcus granulosus. Then we have vectors. Vectors are arthropods that transmit parasites to hosts. Vectors can be biological or mechanical. Biological vectors are those in which the parasite multiplies. For example, the tsetse fly is the biological vector for Trypanosoma brucei, and the mechanical vector is the one in which the parasite does not multiply. These are arthropod vectors that mechanically transfers the infective form of the parasite from one host to another. For example, we have the house flies which act as mechanical vectors in amoebiasis. They transfer the cysts of Entamoeba histolytica from the infective feces to contaminated food, and humans acquire the infection by consuming such contaminated food. Then we have some parasites which infect human beings as well as animals, and the disease is transmitted from the animals to the humans, and this is called as zoonosis. The animal host is involved in natural life cycle of these parasites, while humans are accidentally involved. For example, we have diseases like Leishmaniasis, South American trypanosomiasis, and African trypanosomiasis. Then coming to the host-parasite relationship, the relationship between host and parasite can be divided into the following types. Like we have the symbiosis. This is close association, interdependent, and no harm to each other. Okay, no harm to either the host or the parasite. Then we have commensalism. This is association which is not interdependent. The parasite only derives benefit. No harm to each other is there. And parasitism. This is an association which is again not interdependent. The parasite only derives benefit and in return harms the host. Coming to disease. Disease is a clinical manifestation of infection which shows the active presence and replication of the parasite causing damage to the host. It may be mild, severe, and fulminant, and in some cases may be even fatal. Then we have a carrier. A carrier is a person who is infected with the parasite without any clinical disease and who can also transmit the infection to other individuals. Coming to transmission of parasites. Transmission of parasite depends upon the source of infection, the reservoir or the case, and the mode of transmission. Coming to the source of infection. So we have humans as the source of infection. So man is the source or reservoir for majority of parasitic infections. For example, amoebiasis. The infection is transmitted from one infected person to another, and this is called as anthropogenosis. Then we have animals. The infection which is transmitted from infected animals to humans is called as zoonosis. We've just seen this. And the infection can be transmitted to humans either directly or indirectly through vectors. For example, we have echinococcosis, which is transmitted from the dogs, and toxoplasmosis transmitted from cats to humans. Coming to vector, vector is an agent, usually an arthropod, which transmits the infection from one infected human being to another. Vector can be biological or mechanical, as we've discussed earlier. An infected blood-sucking insect can transmit the parasite directly into the blood circulation during its blood meal. For example, in case of Plasmodium, which causes malaria. Then coming to contaminated soil and water. So soil contaminated with human feces containing eggs of the parasite can act as an important source of infection. For example, in case of the roundworm, the Ascaris species, and Trichuris trichura. Then water. Water which is contaminated with human feces can also be responsible for causing infections, like in case of Entamoeba histolytica and Giardia lamblia. The contaminated water needs to be ingested. The next coming to raw or undercooked food. Raw beef containing the larvae of Cysticercus bovis and pork containing Cysticercus cellulosae are some of the examples of infections which are transmitted through consumption of raw or undercooked meat. And then we have other sources of infections like the fish, crabs, and aquatic plants. The infective forms of parasites may be transmitted from one host to another by following ways, like oral or fecal-oral route. It's the most common mode of transmission of parasites. The infection is transmitted orally by ingestion of food, water, or vegetables contaminated with feces containing the infective stages of the parasites. For example, cysts of Entamoeba histolytica contaminate the food and water which is ingested. Next, coming to the modes of transmission, then we have the next like penetration of the skin and mucous membrane. Infection is transmitted by the penetration of the larval forms of the parasite through the unbroken skin or through the intact skin. For example, the filariform larvae of Strongyloides, stercoralis, and hookworm, which can penetrate through skin of an individual walking barefooted over fecally contaminated soil, or by introduction of the parasite through blood-sucking insect vectors, for example, the Plasmodium species. Then, parasitic infections can also be transmitted sexually, for example, in case of Trichomonas vaginalis. Then, parasitic infections can also be transmitted by bite of vector. Like many parasitic diseases, for example, malaria, which is transmitted by female Anopheles mosquito, filaria, transmitted by Culex mosquito, leishmania, transmitted through sandfly, Chagas disease through redouet bug, and African sleeping sickness through tsetse fly. Then we have vertical transmission, transmission from mother to the fetus, for example, in case of Toxoplasma gondii. Then we have blood transmission of parasitic infection through blood transfusion, for example, in cases of Plasmodium. If a person is having infection and the person donates blood, then in such cases the infection can be transmitted from one person to another. Next, coming to autoinfection, few intestinal parasites may be transmitted to the same person by contaminated hands or by reverse peristalsis. It is observed in Cryptosporidium parvum, Tinea sodium, Enterobius vermicularis, Strongyloides stercoralis, and Hymenopus nana infections. Coming to the life cycle of parasites, the life cycle of any parasite may be either direct or indirect. A direct life cycle is also called as a simple life cycle, and in this type of life cycle, only one single host is required for completion of the life cycle. For example, we have Entamoeba histolytica, Giardia, Lamlia, then we have helminths like H. nana, Ascaris lumbricoides, which require only one single host for completing their life cycles. Then we have indirect life cycles, which is also called as a complex life cycle. In this type of life cycle, the parasites require two different hosts for completing the life cycle. Some of the helminths also require three hosts, one definitive and two intermediate hosts for completing of the life cycle. For example, we have Leishmania species, which requires man as a definitive host and sandfly as an intermediate host. We can have many more examples of such parasites which require two different hosts for completion of its life cycle. Let's go through these slides for a moment. These are the examples of parasites which require three hosts, one definitive and two intermediate for completion of life cycle. For example, Diphyllobothrium species, where man is the definitive host, Cyclope is the first intermediate host, and fish is the second. Likewise, we may have many more such examples like Fasciola hepatica, Fasciola vascae, Paragonimus species, Clonorchus sinensis, etc. Coming to the pathogenesis of parasitic diseases, these parasites can cause damage to humans by many other ways, like we have the different mechanisms of trauma. Like for example, we have eggs. The trematode eggs are large in size and can be deposited inside the intestinal mucosa. Like in case of Cystosoma mansoni, they can be deposited in the bladder, like in case of Cystosoma hematobium, or in the lungs, like we have Paragonimus vestimini, in the liver, like Pasteurella hepatica, and they cause mechanical irritation. Then we have the larvae of certain parasites, migration of several helminthic larvae, for example, the hookworms and strongyloides or ascaris in the lungs produce traumatic damage of the pulmonary capillaries, which lead to pneumonitis. Then we have the adult worms of the parasites. The adult worms of parasites like hookworms, strongyloides, ascaris, and tinea, they get adhered to the intestinal wall and cause mechanical trauma or mechanical obstruction. Then they cause space occupying lesions, like certain parasites produce characteristics cystic lesions that may compress the surrounding tissue or organs, for example, in hydrated cyst caused by Echinococcus granulosus. Then they may cause inflammatory reaction. Many of these parasites induce cellular proliferation and infiltration at the site of their multiplication. For example, we have Endamoeba histolytica, which provokes inflammation of the large intestine, leading to the formation of amoebic granuloma. Then these parasites can also produce certain enzymes and certain lytic enzymes. The obligate intracellular parasite of man, like Plasmodium leishmania and Trypanosoma, produce several enzymes which cause digestion and necrosis of the host cells. For example, we have Entamoeba histolytica, which produces various enzymes like the cysteine proteinases, hydrolytic enzymes, and amoebic pore-forming proteins that lead to the destruction of the target tissues. And these parasitic forms can also produce certain toxins. Toxins are produced by a few of them. These toxins are responsible for the pathogenesis of the disease. For example, we have Entamoeba histolytica, which causes different toxins. Then we have allergic manifestations. These allergic manifestations are seen in infected hosts due to many metabolic and excretory products caused or products of these parasites. And these products get absorbed in the circulation, which ultimately leads to various allergic manifestations or hypersensitivity reactions in the sensitized hosts. For example, we have cystosoma, which causes cercardial dermatitis. Then rupture of hydrated cyst, which leads to anaphylactic reaction. The hydrated cyst consists of a component or a liquid which is allergic. Then we have neoplasias. Some of the infections can contribute to the development of neoplasia. For example, in cases of cystosoma hematobium, which causes bladder carcinoma. Then Clonorchis sinensis and Opistropis causes cholangiocarcinoma. Then we have secondary bacterial infections. These can be seen in some of the helminthic infections. For example, cystosoma and strongyloides. So basically, there is a parasitic infection which gets secondarily infected by bacteria. Then coming to the immune response in parasitic diseases. So the immune response depends on two factors, the host factor as well as the parasite factors. The host factor, like the immune status of the host, age, any underlying disease, then nutritional status of the host, genetic constitution, and various other mechanisms of the host govern the immune response given by the host against the parasitic infection. And the parasitic factors constitutes the size, route of entry, frequency of infection, parasitic load, and various immune evasion mechanisms by the parasite. Broadly, the host immunity may be of the two types, the protective immune response and the harmful immune response. The protective immune response is in the form of innate as well as acquired immunity, and the harmful immune response or the unwanted immune response is in the form of hypersensitivity reactions. Coming to the protective immune response, both innate and acquired immunity plays an important role in protecting the host. Some infections are removed completely, while few are difficult to eliminate. In some infections, the host immune response is sufficient to resist further infection but not able to clear or destroy the parasite completely. The immunity lasts till original infection remains active and prevents further infection. And this is known as infection immunity or primulation, or concomitant immunity or incomplete immunity. This is seen in cases of malaria, schistosomiasis, schwannosis, toxoplasmosis, and Chagas disease. Then coming to innate immunity, this is the immunity which exists by virtue of the genetic makeup or genetic constitution of the host. There are certain factors which influence the innate immunity, like the age. The two extremes of ages have low immunity. For example, very small children and very aged people will have suppressed or low immunity, and hence they will be more prone or vulnerable to parasitic infections. Then the gender of a person. Certain diseases are seen predominantly in male than the females. Then the nutritional status of the host. If, of course, the nutritional status is poor, the person is more prone to infection. Malnourished people are more vulnerable to infections. And the genetic makeup of the host. So, for example, sometimes people with sickle cell disease are uh, resistant to malaria. Okay. Then coming to the components of innate immunity, we have the anatomic barriers like the skin and the mucosa. Skin is an important barrier for the parasite that enters by cutaneous routes like cystosoma, hookworms, and strongyloides. And we have physiological barriers. This includes temperature, pH, and various other substances like the lysozymes, interferons, and complement. Then we have phagocytosis. Phagocytic cells like the macrophages and macrophages act as the first line defense mechanisms in parasitic infections. Then complement system is present in the blood, which will kill the extracellular parasites by forming the membrane attack complexes. Then we have the natural killer cells. These are important in killing the helminthic parasites. Next, coming to the acquired immunity. So, acquired immunity is the resistance offered by an individual during the resistance acquired by an individual during its lifetime, following exposure to the specific infectious agent. This is not by virtue of the genetic makeup or birth of the individual. So, this is mediated by production of antibodies by the B cells or by the T cells in the form of certain cytokines. So we have the B cells which will produce antibodies, and we have the T cells which can produce certain cytokines like the interleukins and the interferons, which act against the parasites. And then we have the unwanted or harmful immune response. Sometimes the immune response is harmful, and this is called as the hypersensitivity reaction. Reactions, there are 4 types of hypersensitivity reactions: type 1, 2, type 3, and type 4. And all of these will play a role in parasitic diseases. Coming to the laboratory diagnosis of parasitic infections. So parasitic infections can be diagnosed microscopically or macroscopically. So macroscopically is usually by observing the external characteristics of the specimens. For example, sometimes we need to collect the freshly passed feces of the patient, and this feces is macroscopically observed for presence of blood mucus, like in case of amoebic dysentery. Then we can look for the parasitic worms. For example, in cases of Ascaris lumbricoides, the adult worms can be seen with the naked eyes. Then we have the microscopic observation, so we can make wet mounds of the specimens of stool and observe under the microscope to see different forms of the parasites, like we can see the cysts, we can see eggs, we can see larvae of the parasites in the specimen. Then we can also culture the parasites. But culturing parasite requires sophisticated technology, and it also requires a very expert hand. So we do not follow these techniques. We do not culture parasites on for routine diagnosis. Of course, they can be done for research purpose. Then we have immunodiagnosis. We can identify or detect certain antigens and antibodies against the parasites by different serological tests, like mostly we have the ELISA. Excuse me. Then we have certain intradermal skin tests. Okay, we can inoculate the antigens of the parasites in the skin and look for certain reactions. Then we have molecular methods. We have xenodiagnosis. Xenodiagnosis is diagnosis in the vectors. The specimen is inoculated in the vector and we observe the growth of the parasite in the vector. And we may have animal inoculations. Laboratory animals can be inoculated. And we have the imaging techniques like the MRI, CT scans, etc. Coming to the treatment of parasitic diseases. Treatment of parasitic diseases is primarily based on the chemotherapy. Antiparasitic drugs are available, and in some cases surgery may also be required. Various antiparasitic drugs are used for the treatment and prophylaxis of parasitic infections. We can go through these slides. So we have a huge range of antiparasitic drugs which can be used against different parasites. So you can just go through these slides for a moment. So we have a range of antiparasitic drugs like metronidazole, chloroquine, paromomycin, amphotericin B, etc. Okay. So with this, we end up with the session which discusses introduction to parasites or introduction to parasitology. So today in the discussion, we have gone through the important terminologies used in parasitology. We have seen what are the different life cycles, what are the diseases caused by these different parasites, and what are the different treatment measures those can be taken against these parasitic infections. What are the different laboratory diagnostic methods? Now, when we'll be doing the individual parasite, we'll be seeing each of these aspects into details for each of the particular parasite. So with this, we end up with the discussion of introduction to parasitology. Thank you.